Diverse Aspects of DNA, From Genomic Structure to Functional Roles, by Sir Noliboy Ramabantanos. The origin of DNA is a complex scientific question. DNA, or deoxyribonucleic acid, is a molecule that contains the instructions an organism needs to develop, live, and reproduce. The prevailing scientific understanding is that DNA likely originated on Earth through a process of chemical evolution, possibly in the primordial soup of early Earth's oceans. However, the exact details of how life and DNA originated remain areas of active research and exploration in the field of abiogenesis. DNA is composed of 4 nucleotide building blocks: adenine, A, thymine, T, cytosine, C, and guanine, G. These nucleotides are arranged in a double helix structure, where A pairs with T and C pairs with G. The sequence of these nucleotides carries genetic information. DNA also contains a sugar phosphate backbone that provides structural support for the molecule. The entire structure of DNA is often described as a twisted ladder or double helix. Mathematically, DNA is often represented using the 4 letters corresponding to its nucleotide bases: A, T, C, and G. In computational biology and bioinformatics, these letters are used to create sequences that can be analyzed and compared. Algorithms and mathematical models are employed to study DNA sequences, analyze genetic data, and understand biological processes such as gene expression, mutation patterns, and evolutionary relationships. Additionally, concepts from information theory, such as entropy, are applied to quantify the information content in DNA sequences. There are several types of DNA, each serving different purposes within living organisms. Genomic DNA. This is the complete set of genetic material present in a cell or organism. It includes both coding, genes, and non-coding regions. Coding DNA. This portion of genomic DNA contains genes that provide instructions for building proteins and other functional molecules. Non-coding DNA. This part does not code for proteins but may have regulatory functions, structural roles, or other as yet undiscovered purposes. Mitochondrial DNA, mtDNA. Found in the mitochondria, these small circular DNA molecules are inherited maternally and are involved in energy production within cells. Chloroplast DNA. Present in the chloroplasts of plant cells, it plays a role in photosynthesis. Like mtDNA, it is also circular. Plasmid DNA. Small circular pieces of DNA separate from the chromosomal DNA, often found in bacteria. They can carry accessory genes, including those providing antibiotic resistance. cDNA, complementary DNA. Synthesized from messenger RNA, mRNA. It represents the coding regions of genes and is often used in molecular biology research. Satellite DNA. Comprising short, repetitive sequences, satellite DNA is found near centromeres and is involved in chromosome structure. Understanding these various types of DNA is crucial for unraveling the complexities of genetic information and its diverse functions. Genomic DNA refers to the entire complement of genetic material within the cells of an organism. It serves as the blueprint for the development, function, and regulation of that organism. Let's delve into the details. Composition. Genes. These are specific sequences of DNA that encode instructions for building proteins, which are crucial for the structure and function of cells. Genes are considered the functional units of genomic DNA. Non-coding regions. These are stretches of DNA that do not directly code for proteins. They were once considered junk DNA, but research has revealed that non-coding regions play important roles in gene regulation, chromosome structure, and other cellular processes. Organization. Chromosomes. Genomic DNA is organized into structures called chromosomes. In humans, for example, there are 23 pairs of chromosomes in each cell's nucleus. Structural genes. These are genes that code for proteins and are involved in the physical structure and function of cells and tissues. Regulatory elements. Non-coding regions often contain regulatory elements like promoters, enhancers, and silencers. These elements control when and to what extent genes are expressed, transcribed into RNA, and translated into proteins. Function. Genetic information. Storage. Genomic DNA serves as a repository of genetic information, containing the instructions necessary for the organism's growth, development, and functioning. Transmission of traits. Genomic DNA is passed from one generation to the next during reproduction, ensuring the inheritance of traits from parents to offspring. Variability. Genetic variation. Genomic DNA exhibits variation between individuals of the same species. This variation is the basis for diversity among organisms and is crucial for adaptation to changing environments. Technological applications. Genome sequencing. Advances in technology have enabled the sequencing of entire genomes, providing a comprehensive understanding of an organism's genetic makeup. Genomic medicine. Studying genomic DNA allows for insights into genetic disorders, personalized medicine, and targeted therapies based on an individual's genetic profile. In summary, genomic DNA is a complex and dynamic molecular entity that contains both coding and non-coding regions, orchestrating the intricacies of life through the transmission and expression of genetic information. Understanding genomic DNA is fundamental to unraveling the mysteries of genetics and advancing fields like genomics and personalized medicine. Coding DNA refers to the specific segments of genomic DNA that contain the information necessary to synthesize proteins and other functional molecules. These regions, known as genes, play a fundamental role in determining the structure and function of cells. Let's delve into the details. Gene structure, exons and introns. Coding DNA is organized into exons, coding regions, and introns, non-coding regions. During a process called splicing, introns are removed and exons are joined together to form a mature mRNA, messenger RNA transcript. Protein synthesis, transcription. The first step in protein synthesis involves the transcription of a gene. RNA polymerase reads the coding DNA strand and synthesizes a complementary mRNA strand. Translation: the mRNA carries the genetic code from the nucleus to the ribosomes in the cytoplasm. Ribosomes then read the mRNA sequence in sets of 3 nucleotides, codons, and facilitate the synthesis of a corresponding amino acid chain, which folds into a functional protein. Functional significance. Proteins as building blocks. The proteins synthesized from coding DNA are essential building blocks of cells and tissues. They contribute to the structural integrity and perform various functions within the organism. Enzymes and catalysts. Many proteins act as enzymes, facilitating biochemical reactions and serving as catalysts for metabolic processes. Transport and signaling. Proteins play crucial roles in cellular transport, signal transduction, and communication between cells. Gene regulation, promoters and enhancers. Coding DNA contains regulatory elements, such as promoters and enhancers, which control the initiation and level of gene expression. Transcription factors. Proteins called transcription factors bind to specific sequences in the coding DNA, regulating the rate of transcription and influencing the synthesis of proteins. Variability and mutations. Genetic variation. Variations in coding DNA contribute to genetic diversity among individuals within a population. Mutations. Changes in the coding DNA sequence, known as mutations, can lead to altered protein structure or function. Some mutations may have no impact, while others can contribute to diseases or provide evolutionary advantages. Genomic technologies. CRISPR-Cas9. Emerging technologies like CRISPR-Cas9 allow scientists to modify specific sequences within coding DNA, offering possibilities for gene editing and therapeutic interventions. In summary, coding DNA is a critical component of genomic DNA, providing the instructions for the synthesis of proteins that form the molecular basis of cellular structure and function. Understanding coding DNA is essential for unraveling the complexities of genetic processes, molecular biology, and the development of targeted therapies in medicine. Non-coding DNA constitutes a significant portion of the genomic DNA that does not directly encode proteins. Despite being initially labeled as junk DNA, research has unveiled the crucial roles non-coding DNA plays in various cellular processes. Let's explore the details. Types of non-coding DNA. Promoters and enhancers. These regulatory elements are involved in controlling the initiation and level of gene expression. They are crucial for the binding of transcription factors and the regulation of nearby genes. Introns. Non-coding regions found within genes that are transcribed but not translated into proteins. Introns are often spliced out during the processing of mRNA. Intergenic regions. Areas between genes that do not contain protein coding sequences. These regions may have regulatory elements influencing nearby genes. Regulatory functions: microRNAs, miRNAs, and small RNAs. Non-coding RNAs can regulate gene expression by binding to mRNA, preventing translation, or promoting mRNA degradation. Long non-coding RNAs, lncRNAs. These longer non-coding transcripts play diverse roles, including regulating chromatin structure, gene expression, and acting as molecular scaffolds. Structural roles: telomeres, non-coding DNA at the ends of chromosomes that protects them from degradation. Telomeres play a crucial role in maintaining chromosomal integrity during cell division. Centromeres, non-coding DNA regions essential for proper chromosome segregation during cell division. Epigenetic regulation, DNA methylation. Non-coding DNA regions can undergo methylation, a chemical modification that regulates gene expression by influencing the accessibility of the DNA to cellular machinery. Histone modification. Non-coding DNA regions are involved in the modification of histone proteins, affecting chromatin structure and gene expression. Evolutionary significance. Pseudogenes, non-coding DNA sequences resembling functional genes, but are non-functional due to mutations. They may have evolutionary significance. Transposons and retroelements. Non-coding DNA includes mobile genetic elements that can move within the genome, influencing genetic diversity and evolution. Emerging discoveries. Functional non-coding elements. Ongoing research continues to uncover the functions of previously unrecognized non-coding elements, revealing their importance in cellular processes. Technological advances. Functional genomics. Techniques like CRISPR screening allow scientists to systematically study the functions of non-coding regions and understand their roles in cellular processes. In summary, non-coding DNA is a diverse and essential component of the genome, with regulatory, structural, and yet-to-be-discovered functions. Appreciating the complexity of non-coding DNA is crucial for understanding the intricacies of gene regulation, cellular function, and the broader landscape of genomics. Mitochondrial DNA, mtDNA, is a unique and essential component of cellular genetic material located within the mitochondria, the energy-producing organelles in eukaryotic cells. Let's delve into the details. Structure and characteristics. Circular DNA. Unlike the linear DNA found in the cell nucleus, mtDNA is a small circular molecule. This circular structure is reminiscent of bacterial DNA, supporting the endosymbiotic theory suggesting that mitochondria evolved from free-living bacteria engulfed by ancestral eukaryotic cells. Maternal inheritance. One distinctive feature of mtDNA is its exclusive inheritance from the mother. During fertilization, the mitochondria in the sperm are usually excluded, and only the mitochondria from the egg contribute to the mitochondria in the offspring. Genetic content. Limited coding genes. mtDNA encodes a small number of essential genes, primarily involved in the production of proteins related to the electron transport chain and oxidative phosphorylation, which are crucial for energy production. Genetic variation. Despite its limited coding capacity, mtDNA is subject to genetic variation, and mutations in mtDNA can have significant implications for cellular function and health. Energy production. Oxidative phosphorylation. Mitochondria play a central role in cellular respiration, a process that generates ATP, adenosine triphosphate, the cell's primary energy currency. The electron transport chain embedded in the inner mitochondrial membrane utilizes proteins encoded by mtDNA to generate ATP through oxidative phosphorylation. Mitochondrial disorders. Maternal inheritance of diseases. Since mtDNA is maternally inherited, mitochondrial disorders caused by mutations in mtDNA are passed from mother to offspring. These disorders often affect tissues and organs with high energy demands, such as muscles and the nervous system. Leber's hereditary optic neuropathy, LH-ON, and mitochondrial myopathies, examples of disorders associated with mtDNA mutations. Evolutionary perspectives. Endosymbiotic theory. The presence of mtDNA in mitochondria supports the endosymbiotic theory, which posits that mitochondria originated from ancient symbiotic bacteria engulfed by ancestral eukaryotic cells. Forensic and anthropological applications. Maternal lineage tracing. Due to its maternal inheritance, mtDNA is used in forensic science and anthropology to trace maternal lineages and study population genetics. Technological advances. Mitochondrial replacement therapy. A technique designed to prevent the transmission of mitochondrial disorders by replacing defective mtDNA with healthy donor mtDNA. In summary, mitochondrial DNA is a crucial component involved in energy production within cells, with unique characteristics such as circular structure, maternal inheritance, and a limited set of coding genes. Understanding mtDNA is essential for comprehending cellular function, inheritance patterns, and the evolutionary history of eukaryotic cells. Chloroplast DNA is the genetic material found within the chloroplasts of plant cells, and it is crucial for the process of photosynthesis. Similar to mitochondrial DNA, mtDNA, chloroplast DNA has unique characteristics, including its circular structure. Let's explore the details. Chloroplast structure. Organelle. Location. Chloroplasts are membrane-bound organelles found in plant cells and some other eukaryotic organisms. They are responsible for photosynthesis, the process by which plants convert light energy into chemical energy. Double membrane. Chloroplasts have a double membrane structure, and their inner membrane surrounds a semi-fluid matrix called the stroma, where the circular chloroplast DNA is located. Characteristics of chloroplast DNA. Circular DNA. Similar to bacterial and mitochondrial DNA, chloroplast DNA is circular. This circular structure is a relic of the endosymbiotic theory, suggesting that chloroplasts originated from free-living photosynthetic bacteria, engulfed by ancestral eukaryotic cells. Multiple copies. A single chloroplast can contain multiple copies of the circular DNA molecule. Role in photosynthesis. Photosynthetic machinery. Chloroplast DNA encodes essential genes that contribute to the photosynthetic machinery. These genes play a role in the synthesis of proteins involved in capturing light energy, converting it into chemical energy, and producing glucose. Thylakoid membranes. Chloroplasts contain thylakoid membranes where the light-dependent reactions of photosynthesis occur. Proteins encoded by chloroplast DNA are integral to the structure and function of these membranes. Genetic variation. Plastid inheritance. Chloroplast DNA is generally maternally inherited in plants, similar to mtDNA in animals. However, exceptions exist, and in some cases, paternal chloroplast inheritance has been observed. Genetic diversity. Variations in chloroplast DNA sequences contribute to genetic diversity within plant populations. Endosymbiotic theory. Evolutionary significance. The presence of circular DNA in chloroplasts supports the endosymbiotic theory, suggesting that chloroplasts originated from ancestral photosynthetic bacteria that were engulfed by eukaryotic cells. Plastid types. Plastid diversity. Chloroplasts are a type of plastid, and plant cells may contain different types of plastids with distinct functions, such as chromoplasts, responsible for pigment synthesis, and amyloplasts, involved in starch storage. Biotechnological applications. Genetic engineering. Manipulating chloroplast DNA is of interest in genetic engineering for improving crop traits, increasing photosynthetic efficiency, and developing transgenic plants. In summary, chloroplast DNA is a vital component for the functioning of chloroplasts and the process of photosynthesis in plant cells. Its circular nature, similar to mitochondrial DNA, highlights the evolutionary history of endosymbiosis and the integration of once independent organisms into the cellular machinery of eukaryotes. Plasmid DNA is a small, circular, extrachromosomal piece of DNA that is separate from the chromosomal DNA. Plasmids are commonly found in bacteria and some other types of microorganisms. Let's explore the details. Structure of plasmid DNA. Circular structure. Plasmids are typically small, circular DNA molecules. This circular shape distinguishes them from the linear chromosomal DNA found in the bacterial nucleus. Double-stranded DNA. Plasmids consist of double-stranded DNA, and they replicate independently of the chromosomal DNA. Origins and distribution. Bacterial cells. Plasmids are prevalent in bacteria and can be found in various species. They are not limited to specific bacterial genera, and are often shared among different bacteria through processes like horizontal gene transfer. Archaea and eukaryotes. While less common, plasmids have also been identified in archaea and some eukaryotic microorganisms. Functions and features. Accessory genes. Plasmids often carry accessory genes that are not essential for the basic functions of the host organism, but can provide additional advantages. Antibiotic resistance. One of the well-known functions of plasmids is carrying genes that confer resistance to antibiotics. Bacteria with plasmids containing antibiotic resistance genes can survive exposure to antibiotics. Toxin production and virulence. Some plasmids carry genes encoding toxins or proteins that enhance the virulence of the bacteria, allowing them to infect host organisms more effectively. Replication and maintenance. Autonomous replication. Plasmids have their own origin of replication, enabling them to replicate independently of the chromosomal DNA. This autonomy ensures their presence in daughter cells during cell division. Partitioning systems. Plasmids often possess partitioning systems that help ensure their proper distribution to daughter cells during cell division. Conjugation and horizontal gene transfer. Conjugation. Bacteria can transfer plasmids horizontally to neighboring bacteria through a process called conjugation. This allows the rapid spread of beneficial traits, such as antibiotic resistance, within bacterial populations. Transformation and transduction. Plasmids can also be transferred via other mechanisms, such as transformation, uptake of extracellular DNA, and transduction, transfer of genetic material by viruses. Biotechnological applications. Gene cloning. Plasmids are widely used in genetic engineering for gene cloning. Scientists can insert a gene of interest into a plasmid, which is then introduced into a host organism for replication and protein production. Recombinant DNA technology. Plasmids are essential tools in recombinant DNA technology, facilitating the manipulation and transfer of genes between organisms. Environmental and clinical implications. Spread of antibiotic resistance. Plasmids carrying antibiotic resistance genes contribute to the spread of antibiotic resistance in bacterial populations, posing challenges in clinical and environmental settings. In summary, plasmid DNA is a versatile genetic element, commonly found in bacteria, that plays a crucial role in various biological processes, including the spread of accessory genes and the development of antibiotic resistance. Understanding plasmids is essential in both basic microbiology and biotechnological applications. cDNA, complementary DNA, is a synthetic DNA molecule that is generated from a messenger RNA, mRNA template. It is a crucial tool in molecular biology research, particularly in studying gene expression and cloning functional genes. Let's explore the details. Synthesis of cDNA. Reverse transcription. cDNA is synthesized through a process called reverse transcription. This involves using the enzyme reverse transcriptase to convert mRNA into a complementary DNA strand. The resulting cDNA molecule is single-stranded. Purpose and applications. Gene expression studies. cDNA is employed to analyze and quantify gene expression levels. By reverse transcribing mRNA into cDNA, researchers can study the transcripts of specific genes. Cloning and recombinant DNA technology. cDNA is often used in cloning functional genes. It allows the production of a DNA copy that lacks introns, non-coding regions, present in genomic DNA, representing only the coding regions, exons, of a gene. Library construction. cDNA libraries are created by cloning cDNA molecules into vectors, such as plasmids or viruses, providing a resource for researchers to study the transcriptome of a particular tissue or organism. Advantages of cDNA. Focus on coding regions. cDNA specifically represents the coding regions of genes, excluding non-coding introns. This focus is advantageous for studying protein-coding genes and their expression cDNA. Single-stranded nature. cDNA is usually synthesized as a single-stranded molecule, making it compatible with various molecular biology techniques. cDNA libraries. Creation. cDNA libraries are collections of cloned cDNA fragments representing the genes expressed in a particular cell, tissue, or organism at a specific time. Analysis of transcriptome. Researchers can use cDNA libraries to analyze the transcriptome, which is the complete set of RNA molecules produced by the genome. Real-time PCR, qPCR. Quantification of gene expression. cDNA is commonly used as a template in quantitative polymerase chain reaction, qPCR, to quantify the amount of mRNA for specific genes, providing insights into gene expression levels. Differential display and race. Rapid amplification of cDNA ends. Differential display. cDNA is employed in techniques like differential display to compare gene expression patterns between different experimental conditions. Race. cDNA is used in race to amplify the 5 and 3 ends of a specific mRNA, facilitating the identification of full-length transcripts. Single-cell analysis researchers. Single-cell cDNA sequencing. Advances in technology allow the generation of cDNA from single cells, enabling to analyze gene expression at the single-cell level. Limitations. Incomplete information. cDNA represents the mRNA present in a cell at a specific moment, providing information about expressed genes, but not about non-coding RNA or potential splicing variants. In summary, cDNA is a versatile tool in molecular biology, enabling researchers to study gene expression, clone functional genes, and create valuable resources like cDNA libraries. Its synthesis from mRNA serves as a critical step in understanding the transcriptome and unraveling the complexities of gene regulation and function. Satellite DNA refers to a class of repetitive DNA sequences characterized by short tandem repeats, repeating units of a few nucleotides, arranged in a head-to-tail fashion. These sequences are often found near the centromeres and play a role in the structure and function of chromosomes. Let's explore the details. Repetitive nature. Short tandem repeats, STRs. Satellite DNA consists of short sequences, usually 5 to 100 base pairs long, repeated in a tandem array. Highly repetitive. The repetitive nature of satellite DNA means that these sequences are repeated many times within a specific region of the chromosome. Location near centromeres. Centromeres. Satellite DNA is commonly found in the vicinity of centromeres, the specialized regions of chromosomes essential for proper segregation during cell division, mitosis and meiosis. Heterochromatin formation. Satellite DNA is often associated with heterochromatin, the condensed and transcriptionally inactive form of chromatin. This contributes to the structure and function of the centromere. Functions and roles. Centromere function. Satellite DNA is critical for the formation and maintenance of functional centromeres. It provides a platform for the assembly of kinetochore proteins, which are essential for chromosome segregation during cell division. Structural support. The repetitive nature of satellite DNA contributes to the physical structure and stability of the centromere region, ensuring proper attachment of microtubules and equal distribution of chromosomes to daughter cells. Types of satellite DNA. Mini-satellite DNA contains longer repeat units, tens to hundreds of base pairs, and is often associated with variable number tandem repeats, VNTRs. Microsatellite DNA comprises shorter repeat units, usually 1 to 6 base pairs, and is associated with short tandem repeats, STRs, or simple sequence repeats, SSRs. Variability and polymorphism. Population variation. The number of repeats in satellite DNA can vary among individuals, contributing to genetic diversity within a population. Forensic applications. Variability in satellite DNA is utilized in forensic DNA analysis for individual identification, especially in the case of microsatellite DNA, where variations in repeat numbers are assessed. Evolutionary significance. Rapid evolution. Satellite DNA sequences can evolve rapidly, and variations in repeat numbers can arise relatively quickly. This rapid evolution is thought to be linked to the dynamic nature of centromeres and their role in chromosome segregation. Technological applications. Cytogenetics. Satellite DNA is used in cytogenetics to study chromosomal structure and identify specific chromosomes. Fluorescence in situ hybridization. FISH. In FISH, satellite DNA probes can be used to label specific chromosomal regions, aiding in chromosomal identification and structural analysis. In summary, satellite DNA, with its repetitive nature and association with centromeres, plays a crucial role in the structural and functional integrity of chromosomes. Understanding satellite DNA contributes to our knowledge of chromosome dynamics, evolution, and has practical applications in genetics and cytogenetics. All the concepts mentioned, genomic DNA, coding DNA, non-coding DNA, mitochondrial DNA, mtDNA, chloroplast DNA, plasmid DNA, cDNA, and satellite DNA, are considered DNA, deoxyribonucleic acid, because they share key characteristics and functions associated with this fundamental biological molecule, nucleotide structure. All these concepts involve the fundamental building blocks known as nucleotides. DNA nucleotides consist of a sugar, deoxyribose in the case of DNA, a phosphate group, and one of 4 nitrogenous bases, adenine, thymine, cytosine, or guanine. Genetic information storage. DNA is the primary molecule responsible for storing and transmitting genetic information in living organisms. Whether it's the complete set of genetic material in genomic DNA, the coding regions in coding DNA, or the specific genes in cDNA, the common theme is the storage of genetic instructions. Replication. DNA molecules have the ability to replicate, ensuring the transmission of genetic information from one generation of cells or organisms to the next. This is evident in genomic DNA replication during cell division, and the replication of mitochondrial DNA, chloroplast DNA, plasmid DNA, and cDNA in various biological contexts. Genetic variation. Different types of DNA exhibit variability, contributing to the diversity of living organisms. This includes genetic variation within genomic DNA, mtDNA, and chloroplast DNA, as well as the variations observed in plasmid DNA and satellite DNA. Involvement in cellular processes. DNA participates in essential cellular processes. For example, coding DNA provides the instructions for building proteins crucial for cellular function. mtDNA and chloroplast DNA are involved in energy production and photosynthesis, respectively. Plasmid DNA contributes to bacterial resistance and other functions, while satellite DNA plays a role in chromosome structure. Universal presence. DNA is a universal molecule found in all known living organisms. While there are variations in the structure and organization of DNA in different species, the core principles remain consistent. Use of DNA technologies. Various molecular biology techniques and technologies, such as PCR, DNA sequencing, and gene editing, are applicable across these different types of DNA, highlighting their commonality in terms of manipulation and analysis. In summary, the shared features of these concepts, including their nucleotide structure, role in genetic information storage, involvement in cellular processes, and the ability to undergo replication, collectively qualify them as DNA. Despite the diversity in their functions and locations, they all contribute to the fundamental processes that underlie the inheritance and expression of genetic information in living organisms. From the Circle 1111 series of NOLIEISM.